I'm very excited to be here with you guys, and I think we have amazing developments in atopic dermatitis, and now they really are also continuing to alopecia areata. And I'll show you the road we had to pass in atopic dermatitis, and I think we are following perhaps a similar path in alopecia areata. So my disclosures, but I have to say I don't have any patents or any financial gain from any product, and all my grants are coming to Mount Sinai. So atopic dermatitis, all of us are familiar with the disease, but I think for many years, until there was epidemiological data, we didn't know that this is actually the most common inflammatory disease in humans and the most common inflammatory skin disease. New data from Jonathan Silverberg showed 7% of the adults in the United States with atopic dermatitis. About a third of these have moderate to severe disease, so you can imagine how much this impacts general health of the American population. And up to recently, we had a huge unmet need for better treatments to control the disease, particularly long-term, because as dermatologists, yes, we did have phototherapy, oral immune suppressants that are not approved, by the way, besides prednisone that I myself never give to patients with atopic dermatitis. But these are not treatments that we can sustain patients for long-term disease control. And this is what we needed. And due to increased knowledge, the therapeutic drought is finally ending in atopic dermatitis, but it wasn't that simple as it sounds. Now, another important knowledge for you is that atopic dermatitis is the first window, perhaps, to the entire atopic march. All the other atopic conditions follow. Atopic dermatitis initiates in the first 3 months or 6 months of life most of the times, followed by food allergy, allergic rhinitis, and asthma. And I think an important question we need to ask is, can the atopic march be prevented in children that develop already atopic dermatitis by appropriate Th2 targeting? And I think this is the million-dollar question that will be answered probably in the next 5 to 10 years. Now, atopic dermatitis for many years, what prevented therapeutic development is this maybe futile debate, what starts first, the inside out or the outside in, or the immune abnormalities on one hand, or the barrier abnormalities on the other hand. So the inside out, or the immune abnormalities, are characterized by abnormalities in type 2 cytokines, such as IL-4, IL-13, IL-31, but also type 22, such as IL-22. And we also have a vast barrier abnormalities with abnormalities in barrier proteins such as filaggrin, loricrin, lipids, tight junctions, and others. And these allow for penetration of irritants and allergens and microbes, basically ultimately creating the atopic dermatitis phenotype. Now, it's important to note that the type 2 cytokines are increased not only in adults that have chronic disease, but they are increased also in children with very early disease. And this is a collaboration I've done with Amy Paller, known to all of you, that managed to do the incredible GET biopsies in children. And we show together that even the non-lesional skin of children with very early disease, and these are infants within 6 months of disease initiation, they already have increases of IL-13, the primary cytokine of the type 2 immune pathway. And maybe that non-lesional skin of children may be the real disease initiation in children, and the same with IL-31, even more than adults. But the important thing is that type 2 cytokines already are increased very early in the disease, and IL-13 also correlates with the SCORAD. This is a measure of disease severity. Now, important work that was done by my lab, led by Talik Zarnovitsky, that is actually in the audience, showed the same thing in blood. So type 2 inflammation is increased in blood already very early on in infants and in children. So children and infants with they already have systemic type 2 inflammation very early on if they have moderate to severe disease, suggesting the potential need for systemic treatment already very early on in life. Now, we need to also remember important work done in vitro by Donald Leung and others that showed that type 2 cytokines such as IL-4 and IL-13, but also IL-31 was shown, and IL-5 in some studies, alone and together, they are able to inhibit barrier proteins such as filaggrin and loricrin, as you see here, and also they are able to inhibit antimicrobial peptides such as HBD2 and others. And why, you will ask, is this important? Because antimicrobial peptides are important in the setting of infections, to fight infections, and we all know that patients with atopic dermatitis, they have increased Staph aureus colonization and infections, and also they are prone for other infections. Now, important work from Brian Kim, as you know, that I'm so excited. He's joining my department in January. Also linked type 2 cytokines and also JAKs with each, and maybe we can postulate that Th2 cytokines are likely linking skin barrier, immune activation, and itch in patients with atopic dermatitis. Now, we also showed that atopic dermatitis is a heterogeneous disease, much more so than psoriasis, that is much more homogeneous. Atopic dermatitis, depending on the clinical phenotype, such as children with AD, African Americans with atopic dermatitis that have more lichenified lesions, Asians with AD that have more demarcated lesions, or European Americans. So these clinical phenotypes also translate to differences in immune polarization and barrier defects. And these are very important because different phenotypes may need a little bit different therapeutic targeting. We need to remember all of them do share the robust type 2 activation, but some of them may need additional cytokine targeting to really achieve 100% control. Remember, in atopic dermatitis now, we do not have drugs that achieve, like in psoriasis, really 100% control with one cytokine targeting. Now, we all know that type 2 inflammation or atopic conditions are sharing that Th2 activation or type 2 activation in the organs such as skin or other organs, and also the systemic immune activation. And in dermatology, we now have several of them. Beyond dermatology, we have asthma, allergic rhinitis, and others. But now what is emerging is that in the dermatological atopic conditions, alopecia areata is emerging as a disease that becomes part of that atopic march, if you will, of the type 2 inflammation. Another important concept that is also important to alopecia areata is the idea of the systemic inflammation. So in atopic dermatitis, we showed in several studies that patients that have moderate to severe disease with more than 10% body surface area, they have increased systemic inflammation with increased activated T cells, increased circulatory cytokines, including cardiovascular associated markers. And that was exactly at the same time when important epidemiological work from Jonathan Silverberg and others showed in population-based studies increases in cardiovascular disease in our atopic dermatitis patients with a severe disease or moderate to severe disease. And also a work from our group, from Benji Unger, that is now an attending at Sinai, showed using PET-MR increased vascular inflammation in patients with atopic dermatitis that have chronic disease. So having chronic atopic dermatitis is not good for you. It may affect your health later on. And this is also reflected perhaps in the seemingly uninvolved skin, that so-called normal skin that may be a little bit only dry, but it's not as erythematous as the lesional skin. You see, just a little bit flaky. That skin is not normal. It has increases in type 2 cytokines and chemokines such as IL-13 and multiple chemokines of the type 2 immune pathway, and in IL-22 and other molecules. So you already have increases in these immune molecules and also decreases in barrier molecules in that non-lesional skin, perhaps reflecting the idea that these patients need systemic treatment. So how are we testing the contribution of the immune pathways to atopic dermatitis? We are doing it through clinical trials with targeted treatments in AD patients. And I think dupilumab was very instrumental in this field, perhaps showing two things. When you improve the disease with dupilumab, the clinical severity, you inhibit IL-4 receptor, but you also inhibit many other immune abnormalities in atopic dermatitis, and you also inhibit the barrier abnormalities or improve the barrier abnormalities in atopic dermatitis. And this really proved the immune hypothesis of atopic dermatitis and the importance and the relevance of the type 2 immunity to the disease, placing IL-4 and IL-13 as pathogenic cytokines in atopic dermatitis, and really opening the door to all the therapeutic development we are now seeing in atopic dermatitis. So now I want to switch gears and ask, how can this translational revolution that all of us agree is happening in atopic dermatitis also extend to alopecia areata? So when I started, I kind of came to alopecia areata from the back door, like I like to say, because I was focused on atopic dermatitis, and I started to see that some of my patients with atopic dermatitis also have alopecia areata. And that was in a time that only the type 1 immunity was put forward. And I thought that maybe that's not the whole story. And I started to dig in the literature, and I found that there are quite a few publications, and an important one from Abrar Qureshi with more than 2,000 patients while he was at Harvard, showing that atopy in general and eczema in particular is the #1 comorbidity of alopecia areata. And then there are many studies, including a very important population-based study in Israel. You know, in Israel they have a large database and they can do these studies, showing that with each atopic comorbidity, you increase the chance to have alopecia areata. And there are many other studies now. I think it's well accepted now that alopecia areata is part of this atopic comorbidity. And also the largest GWAS study done, published in the GID, actually positioned IL-13 as the highest association, and there are several studies with IL-4 polymorphisms as well. And there are many publications on immune pathways or immune cytokines that are perhaps relevant to atopic dermatitis. Some show Th1 relevance in atopic dermatitis, and Th1 axis is increased indeed in patients with atopic dermatitis, with alopecia areata, and this axis is downregulated with treatments. But recently, the Th2 cytokines also appear to be important in alopecia areata, and also it's clear now that IL-17, IL-22 are completely irrelevant in alopecia areata. Also, there is an IL-17 study that was not successful in alopecia areata. IL-23 is elevated in alopecia areata. We still do not know fully its relevance. But we know that the IL-17 axis is not involved in alopecia areata. We have done a large study, the largest at the time, that compared 30 patients with moderate to severe alopecia areata to patients with atopic dermatitis and psoriasis. And it is important to compare to other diseases because it gives you a sense of how it's positioned compared to the other diseases. And what we showed in these studies is that the Th1 axis is relevant to all of these inflammatory conditions, as you see here, but IL-13, the Th2 cytokine, is only upregulated significantly in alopecia areata and atopic dermatitis. And P19 and P40, the two subunits of IL-23 and IL-12-23, are upregulated again in all of these conditions. Now, importantly, alopecia areata may be a condition where, similar to atopic dermatitis, the actions of cytokines are suppressing hair keratins. And in alopecia areata, you see hair keratins, both early keratins, middle keratins, and late keratins are downregulated, and these keratins need to be upregulated with any successful treatment you have for alopecia areata. There is no such a thing that you improve alopecia areata and you don't upregulate the hair keratins, a little bit similar to atopic dermatitis and the barrier defects. Now, a work from my group showed that in patients with moderate to severe alopecia areata, the inflammation is not only in the scalp, and this was a little bit counterintuitive to us because, right, these patients don't have erythematous lesions, they don't have any visible erythema anywhere, but these patients with moderate to severe disease, they have higher inflammation in blood that is very similar to atopic dermatitis, more than psoriasis, and even a little bit more than atopic dermatitis. Their inflammation in blood is similar to atopic dermatitis with increases in Th2. In Th1, there is some increase in IL-12/23, and interestingly enough, they have increases in markers that are linked to atherosclerosis signaling, right? This is very surprising for alopecia areata. It's a little bit counterintuitive, but I think this is very important, and this is regardless of atopy. So in both patients that have atopy, we know that about a third of patients, if not more, with alopecia areata will have atopy. So both patients without atopy and with atopy had that increased systemic inflammation. Now that is exactly in a similar time when there are several papers, and I'm showing this paper from Korea, in which they showed increased risk of acute myocardial infarction in patients with alopecia areata. And this suggests to us that very similar to atopic dermatitis, you may need to treat early with systemic treatment in patients with alopecia areata. Now, my lab participated in this study from Pfizer with a very unique study in which they took 2 JAK inhibitors with 1 placebo arm. My site also participated in the clinical study. And these are the results of the study, very exciting study. It was one of the first studies that was placebo-controlled in alopecia areata, showing very nice responses. And this drug is now moving forward. When I'm saying this drug, they took 2 drugs, but they are moving forward the JAK3 tech that is called ritalcitinib now. Just one second. And these are the results of the mechanistic study. We presented it in several meetings. We are very happy that now the paper is in press in JAKI. And here we see that the patients the 1600 is the one that is moving forward, ritalcitinib. And here we see that patients that have shorter duration of disease, and this is marked by 3.5 years. So if you have less than 3.5 years, you'll have better chances of success. So it's really important to treat these patients early, as you see. Look at the amazing changes in tissues in the patients, and this is only at 24 weeks, and we know that hair growth takes time, but at only 24 weeks, you really are able to switch the phenotype of patients between week 0 and week 24, particularly in the patients with shorter duration. It's not that the longer duration will not work, but probably it may take more time, and it's a little bit more difficult. So it just tells us that it's important for us to treat our patients with alopecia areata early, and particularly in children. And what we saw also when we looked at different immune pathways is that we obtained modulation of different immune pathways and in hair keratins with both drugs. The Th1 pathway, as you see, the Th2 pathway, IL-12/23, and also in hair keratin. So the hair keratins have to increase, as I told you. Now, another important thing that I want to show you with the drug that is moving forward now in phase 3, at week 24, when we look at what are the correlates between biomarkers and disease severity, the SALT, I want to point out, Th2 biomarkers are at the very top. CCL17, TARC, that's the premier biomarker of the type 2 immune pathway, and IL-13. So that also is suggestive of the type 2 pathogenesis in alopecia areata. Now I want to also show you a little bit from my own investigator-initiated study with dupilumab. This study is published in Allergy, and we are hoping that dupilumab will also become an option for our patients with alopecia areata. These are the inclusion criteria, more than 30% scalp hair loss, and this is a very small study. When you talk about alopecia areata, usually studies are much larger than this. This had 40 patients starting on drug, 20 patients starting on placebo. The patients obtained every week dosing, and after that, they were treated with every week dosing until week 48. At week 48, we put them through the insurance, so some of them did get every week, some of them got only every other week, which can impact the results because, as I told you, these patients have high systemic inflammation and they need perhaps that every week dosing. Another thing to tell you is that at least a third of patients had to have either active AD or a history of AD, and only about 10% of the patients had actually active AD, because usually patients with alopecia areata will not have active AD. But many of them did have, like it happens always if you ask patients with alopecia areata, many of them will have a history of AD or a family history of AD. Surprisingly, and that was surprising to us, many of them had high IgE. They didn't even know about that, but we now see that this is important, and you'll see why this is important. So this is the result of the study. Not impressive data at week 24, because probably this was not the right endpoint that we needed to put. But look at the continuous improvement. It's kind of like a marathon runner. It's not a sprinter in alopecia areata, but I do see it as a potential marathon runner in alopecia areata. And these are the data for SALT 30, SALT 50, and SALT 75. The good thing about alopecia areata, very small placebo responses, even with SALT more than 30. Most of the studies are doing SALT more than 50, and then you even have less placebo responses. But what we discovered is that patients with high IgE and atopy, they have much better responses, and these are about half of the patients. Look at the correlation with high IgE, and IgE, baseline IgE, is able to predict response. And when you look at the group with the high baseline IgE, and this is a very important thing that we will follow up with other studies, then you increase your SALT responses by much. So if you find it's a little bit potentially similar to atopic dermatitis, that if you find the right population, you are able to increase the responses. And you know, I'm a great believer that quality of life is also a very important measure, and this is the correlation between a change in SALT and quality of life of patients. And we know that quality of life measures need to be improved, but it's still highly correlated. And you know, as a clinician, for me, it's important also to see what does it do really to patients. This is a patient, and we have many of these patients in the study, and this is how the patient, and this is real, and I've seen this patient just 2 weeks ago, so it changed multiple lives of multiple patients, and this is a kind of summary, abstract summary from this paper, but I do think that high IgE and atopic background is the secret to this drug in alopecia areata. Thank you so much.